This will be our first lecture on metabolism, and this will be a, a really just an introduction uh, to the basics of some metabolism. And what we're going to cover today will be some bioenergetics and then also reaction types. Right. So the last lecture covered sugars, in particular the more complex sugars, polysaccharides, and glycoconjugates. Okay, today we're going to cover chapter 13. And in chapter 13, uh, what is covered is some bioenergetics, and that kind of leads back to thermodynamics. Hopefully, that will be kind of a review for everyone. Um, and the other thing that's important in chapter 13, it lists reaction types. Okay, and there's really, when you break it down, there's not a whole lot of reaction types that occur in biology, in biochemistry. There's six main types that we're going to talk about. Right? And then the last two sections in chapter 13 cover phosphoryl group transfers, and these are actually very important in metabolism. And then oxidation reduction reactions, redox reactions. Okay, so those are two of the six reaction types, but these are probably stand out a little bit more, at least in metabolism, moving forward. So metabolism is the sum of all chemical reactions in the cell. And here's a chart that shows metabolic pathways. Okay, and it's, as you can see, it's very, very complex. You don't have to memorize all of this, right? That would be very hard to do, very challenging. And to be honest, I work in metabolism doing research, and some of these pathways aren't even really verified experimentally. They're just, sort of thought to occur, or they they found, one of these arrows somewhere up here. They might have found that reaction in let's say yeast, and then they just assume that it occurs in humans. But some of them are really unproven. So even in this day and age, so that just goes to show you how complex metabolism is. Okay. And these pathways are related reactions. Okay. Um, Not going to focus too much on this figure because it's really hard to read, but you have these molecules and then an arrow and then a different molecule, and so that arrow is indicating there's a reaction taking place, and those reactions are catalyzed by enzymes. Okay, and what you'll also notice is that all of these pathways are really interconnected with each other, so it makes kind of like a web. And you'll see that in patients that have mutations in certain enzymes, metabolic enzymes. There'll be a block in one step, and you'll see buildup of products in multiple steps, kind of that branch off from that particular pathway. Okay, so all things are sort of related. Metabolism can be broken down into two parts. Okay, one is what's known as catabolism. Okay, and this is generally thought of as energy producing pathways. Okay, and you're producing energy, and what you're doing with that is you're breaking things down. Okay, so you're taking more complex things. In this example, you have proteins. You're breaking them down into peptides and then into the individual amino acids. Those amino acids can then be broken down further into things like pyruvate, which then gets turned into acetyl CoA and gets fed in the citric acid cycle, which makes energy. Okay. So catabolism is energy producing. Okay, you're taking more complex things and and breaking them down to produce energy. Okay, and you can think of that as catabolic processes. You generally think of things like riding a bike or running. Those are things where you would be breaking things down to get energy out of. Okay. The other part of metabolism is anabolism, and that is where you use energy to form more complex structures. You take less complex structures, building blocks, you put them together, you build up to a more complex structure. And an example is kind of shown here, although that's not maybe not the best example, but you can make amino acids. Here's serine, and then you can go further and connect amino acids together to make proteins, right? And so anabolism or anabolic pathways build up. And one thing you can think of, like macroscopically, anabolic pathways, you can take anabolic steroids to enhance those, and that's why weightlifters or professional wrestlers here have really big muscles, right? They take steroids to get their bodies like that, or at least some of them do. Well, these pathways, these types of pathways, aren't they're connected to each other. They're not standing alone. Okay, so catabolic pathways that are using more complex things, breaking them down into metabolic products to produce energy, in this case in the form of ATP, right? Those, they're not just operating in vacuum, right? Because that ATP is then used in anabolic pathways where you take external nutrients and then you build them up into biopolymers, so things like DNA or proteins. Okay. You're using that energy in the form of ATP that you made in the catabolic pathways. Okay, so these are really you can't have one without the other. Okay. Right, life needs energy. Okay, and if you think back, just in general, living organisms have complex structures. And in biochemistry, the first quarter of the class, you talked about proteins primarily. And this quarter, we've talked about lipids, fatty acids, and membranes. So those are some pretty complex structures, okay? And living organisms really rely on those. Okay? So building complex structures are low in entropy. The only way that's possible is by spending energy. Okay? The ultimate source of energy on the planet is sunlight, okay? And that in turn lets things like plants grow, things that use that sunlight to produce the nutrients they need, and then animals and higher life forms kind of feed off of that. Okay. If we talk about the laws of thermodynamics real quick, just a little review. Living organisms can't create energy from nothing. Okay, that comes from the first law of thermodynamics. The first law of thermodynamics states that energy can't be created or destroyed. It can just be converted from one type of energy to another. Okay. Right, so living organisms can't create energy from nothing. They can't destroy energy into nothing. And they can transform that energy from one form to another. So this in blue is from the first law of thermodynamics. Okay. The second law of thermodynamics states that the entropy of the universe is always increasing. So when we apply that to living organisms, the process of transforming energy, living organisms have to increase the entropy of the universe. Okay. In order to maintain that organization within themselves, right, that's a low entropy state that living organisms have within themselves. they have to be able to extract usable energy from their surroundings and release useless energy in the form of heat back to the surroundings. And that's where you get that increase of entropy for the universe. So remember from thermodynamics that delta G has to be less than 0 for a spontaneous reaction. Okay, and you also have this equation. Hopefully this looks familiar. Delta G equals delta H, which is the enthalpy, minus temperature times delta S, which is the entropy change. So for a process, a given process or reaction, the Gibbs free energy, the change of Gibbs free energy, equals the change in enthalpy minus temperature times the change in entropy. And that has to be negative for a spontaneous process. Okay. And also, if we think about equilibrium, which hopefully again is a review from general chemistry, okay, you can write a particular reaction. This is just a generic reaction. A plus B goes to C plus D. And these lowercase are just the factors, the stoichiometric factors in the reaction. And the double-headed arrow means this is at equilibrium. So we can write an equilibrium expression, or equilibrium constant for that, KEQ, and it's the products multiplied by each other, divided by the reactants. So products over reactants. Okay, you can relate equilibrium and free energy. Okay, and that's done using this equation. Okay, and there's a way to derive this, but you don't have to worry about deriving this. If you remember this equation, we'll be using it, so you should get some practice with it. But the delta G prime naught equals negative RT, R is a constant, T is temperature, times the ln, natural log of KEQ. So the table 13.3 from the book kind of relates this, right? So this equation tells you that the equilibrium constant and delta G are related. Okay. When KEQ is greater than 1, delta G is negative, and therefore the reaction is then spontaneous in the forward direction. If KEQ is less than 1, delta G is going to be positive. That means the equilibrium is going to proceed in the reverse direction. If KEQ is exactly 1, that means delta G naught prime is going to be 0. And that when a reaction has a delta G value of 0, that tells you it is at equilibrium. So actual free energy, the delta G naught prime applies to standard conditions, okay? And that's what really this naught, this superscript 0 implies, that it's standard conditions. So standard conditions are 1 molar of your products and reactants, 1 atmosphere, pH is at 7, and you're at 298 Kelvin. So it's a measure of what direction that reaction is going to go when it's at standard conditions. Okay, but standard conditions don't really apply to real life situations. So an equation for delta G in a real life situation is given here. Okay, the delta G, and again, this delta G is for any condition, okay, is going to be equal to delta G naught prime, that's standard state conditions, plus RT ln of Q. Okay, and Q is the reaction quotient, or it's also known as the mass action ratio. The expression for Q is given here, right? And it looks really familiar, right? It's the same as the KEQ expression. You find it the same way. But the difference is KEQ is at equilibrium. Q is at any conditions. Okay. So Q is the actual conditions, in this case, since we're looking at a chemical reaction, it's the actual concentrations that are that are present. And this is actually a very important equation because even with a reaction that has a positive delta G naught prime, so a reaction that would not be spontaneous at standard state conditions, cells can manipulate the concentrations of the products and the reactants in such a way to make this term cancel out the positive delta G naught prime and get a negative delta G value. So cells can really perform some magic or trickery to get these reactions that normally you would think of as non-spontaneous to get them to go. So let's take a moment and review some concepts from organic chemistry. That was sort of a general chemistry, thermodynamics review we talked about. But when we're looking at metabolism and metabolic pathways, it's also important. We're going to see some basic reaction mechanisms, and I think understanding reaction types, it can be helpful to have some think of it in terms of in organic chemistry terms. So just a quick review of some organic chemistry terms. In biochemistry, most reactions are thermoheterolytic processes. Okay? And what that means is you have either one molecule being split into two or two molecules coming together to form one. Okay? And this happens through the fact that nucleophiles and electrophiles will react with each other. Okay, so remember, nucleophiles are things that things like carbon is a good oxygen are good nucleophiles, things that have unpaired electrons that are fairly electronegative, and electrophiles are things like carbon, things that in particular a carbon with like a double bond to an oxygen, something that's electronegative, that's drawing the electrons away from that carbon then will become fairly electrophilic and be susceptible to a nucleophilic attack. Right? Heterolytic bond breakage often gives rise to transferable groups, and we'll talk about some of those. Protons, phosphates are good transferable groups. Okay. Oxidation of reduced fuels often occurs via transfer of electrons and protons to a dedicated redox cofactor, and we'll talk about some of those. Okay. So going a little bit deeper into this, okay, nucleophiles, right? Things that are, have unpaired electrons, or even things that have a complete negative charge on it. Okay, oxygen is one, sulfur in the form of a sulfhydryl group. Carbanion, we'll see examples of these where the carbon has a negative charge, and then also amine groups that are actually uncharged. Okay, and so not only amines, but things like imidazole. So the histidine side chain has a nitrogen there that can be a nucleophile. Okay. Some electrophiles, as I mentioned, the carbon atom in a carbonyl group, and then there's some others. One of the more important ones is phosphorus in a phosphate group, and we'll see many examples of those. Okay, so the reaction types that we'll see are shown here. Okay, we have ligation, isomerization, hydrolytic or condensation, depending on which way you're going, lyases, group transfer, and oxidation reduction reactions. And probably the two that are the most important moving forward are going to be group transfer and oxidation reduction reactions. So we'll probably see more examples of those, but we do see examples of all of these reaction types in the metabolic pathways we cover in this class. Okay. So energetics. These are just some general terms or general energetics of some of these types of reactions. But you'll probably see in the pathways we look at examples of that are kind of contradictory to this list. So I don't really want to spend a whole lot of time on this, but hydrolysis reactions tend to be strongly favorable or spontaneous by themselves. Isomerization reactions, on the other hand, right, you're just rearranging the same atoms in a molecule. They have smaller free energy changes, so their delta G at standard state values tend to be closer to zero. Complete oxidation of a reduced compound is strongly favorable. Okay, this is how chemotrophs obtain most of their energy. So chemotrophs are living organisms that don't have photosynthesis, basically. In biochemistry, oxidation of reduced fuels with O2 is stepwise and controlled. So on Earth, we have an oxygen atmosphere. Most organisms, at least that we're going to talk about in this class, like us, are aerobic. So we're combining fuel sources with O2. Okay, and that process is stepwise and controlled. Okay, and that's important. You want it to be controlled because if it's kind of out of control, you're not going to get the full benefit of that. Okay. Okay, recall that being thermodynamically favorable is not the same as being kinetically rapid. So certain reactions can be favorable, but that doesn't mean they're fast if they have large activation barriers. Okay, and remember, that's what enzymes do. They lower that activation energy to speed up reactions. Okay. This is a chart in your book, a table just showing some delta G naught prime values for some example reactions. Okay, hydrolysis reactions, again, the general trend is that these are going to be pretty spontaneous, and you can see that they have fairly large delta G values, negative delta G values, and then some isomerization, rearrangement reactions. Those delta G values are going to be closer to zero. Don't need to memorize any of these. You might need to look in this table to answer homework questions if you need to calculate, the delta G of certain reactions. Okay. So let's get into the reaction types in a little bit more detail. Okay, the first group we're just going to call ligations. Okay, and these are examples of nucleophilic carbon-carbon bond formation reactions. Okay, things like the aldol condensation, and here we have a carbanion with nucleophilic attack on this carbonyl carbon. Okay, when that happens, right, the electron mechanism, we watch the electron pushing. Okay, those electrons come out. They can grab a proton and to form an OH group, and now you've created a one molecule from two. Okay, so that's a ligation. You're sticking things together. Okay. Another reaction type from organic chemistry you might have had, right? Now it's an ester, because we have a better leaving group here, so it's going to form this ester instead of an alcohol. Okay. You can have these. Okay, so a ligation, you're sticking things together. These can sort of occur in the reverse direction too. An example of that is a decarboxylation. So you have a CO2 group that can leave a molecule as well. In this case, it's a beta keto acid. Yeah. Second type of reaction is isomerizations, and isomerization is just a rearranging of a molecule. Okay, in this example here, we have glucose 6-phosphate and an enzyme called phosphohexose isomerase changes it to fructose 6-phosphate. This is actually one of the reactions in glycolysis, and we'll be talking more about that in chapter 14. Okay, but all you're doing is you're moving this carbonyl group from position carbon 1 to carbon 2. Okay, so you're switching the carbonyl and the OH group. Okay. Mechanism for how this occurs is shown here. We'll probably get more into this in chapter 14. Okay, so you really not as important to know this right now, but what you have here is a particular amino acid side chain in this enzyme, phosphohexose isomerase, is going to abstract a proton, right? And then you get the electron movement, and you form what's known as an ene-diol intermediate. So you have two OH groups on adjacent carbons and a double bond between those two carbons. Okay, and then that's a very short-lived intermediate, and then it flips back, but in the process it's changing the position of the carbonyl and the alcohol. What you'll notice too is that there's no overall change in oxidation state here, right? You have a carbon double bond oxygen's more oxidized than the carbon single bond with oxygen. So in a sense, this carbon's being more oxidized while this one is not. But the net overall oxidation state of the molecule doesn't change. Okay, the third type of reaction is hydrolysis. And hydrolysis is hydrolysis, meaning lysis meaning break, and hydro meaning water. So you're breaking a bond with water. Okay, so this enzyme sucrase will break the sucrose disaccharide. It will break this disaccharide bond using a water. Okay, and that's shown here. You also have this reaction go in the reverse direction, and that would be forming a bond while eliminating a water. So if we're running this in reverse, this highlighted OH and H will form water. And that's what's known as a condensation reaction. So hydrolysis, breaking a bond with water. Condensation, you're forming a bond and eliminating water from that. Okay. Group 4, lyases. Okay, and these are probably the most difficult to define reaction type. And the definition is breaking a bond or eliminating something without using water, and it's no redox. Okay, so that's kind of a bad example there. But it turns out we actually see many examples of lyase reactions. The reaction that we talked about in was that chapter 12 with signaling, where we have ATP being turned into cyclic AMP by adenyl cyclase. Okay, that's actually a lyase reaction. All right, and we'll point out some more lyase reactions when we go through metabolism. Okay, a lyase reaction, if I was just writing reactions down and asking you, what type of reaction this is, it would be hard for you to, it would probably be hard to kind of pick out, oh, that's a lyase, you know, unless it's a metabolic step that we talked about specifically, that is a lyase reaction. So these are really the hardest one to kind of figure out. Okay. Fifth type are group transfer reactions. Okay, and there's many different things that can be transferred. That can be your group being transferred. Protons are pretty common. Methyl group transfers, acyl group transfers. Okay, that occurs in biosynthesis of fatty acids, which we don't really cover, but we do kind of cover those two carbon groups in the form of an acyl group in metabolism. We will be covering that. Glycosyl transfers, those are attachments of sugar. And then really one of the most important ones that we cover are phosphoryl group transfers. And those are done to activate metabolites, okay, make them into a more high energy, higher energy compound. And so that's kind of shown here, where you have a glutamate and ATP. You're transferring a phosphate from ATP to glutamate to make this a phosphate bound molecule that makes it a higher energy. That phosphate group, then that bond is high in energy. It's a good leaving group, and then you can switch out the amine group there, right? And this is how you can, how the body converts glutamate into glutamine. Okay. So phosphoryl group transfers are going to be pretty important. They're also important in signal transduction. Okay, so things getting phosphorylated. Remember the kinases and the kinase cascades that propagate signals in a cell. Those are using phosphate groups to do that. Okay. And so phosphate group, this group transfer and phosphate in particular, it occurs via a nucleophilic displacement. Okay, so the phosphorus is an sp3 hybridized, and it proceeds like a nucleophilic substitution. Okay, and so this is kind of referred to as SN2-like mechanism from organic chemistry. Okay. So the nucleophile forms a partial bond to the phosphorus, giving it an intermediate that is pentacovalent, right? So there's 5 groups in this intermediate around this phosphate group. Okay. So in this case, Z is another OH group on the phosphate, and W now is something like ADP, where it's accepting that phosphate group. Okay, ATP is frequently the donor of phosphate, and the ATP, we're just going to focus now on the 3 phosphate groups here. It will give up that gamma phosphate, that third phosphate in the chain, to a number of different molecules. Okay. In glycolysis, one of those molecules is glucose. Right. So you can get this nucleophilic attack here by an OH group on glucose. Okay, that'll form a pentacovalent intermediate, and then the phosphate group, because this is a very high energy bond, it will leave with the glucose. ATP, the hydrolysis of ATP is highly favorable under standard conditions. And why is that? Well, there's better charge separation in the products. If you look at this in ATP, these phosphate groups have negative charges. Okay, so you have these negative charges very close to one another. So breaking off that gamma phosphate, you're going to take away some of those negative charges. So you're just simply spreading the charges out, which is favorable. Okay. You have better solvation of the products, so better solution. The water that this is in can interact better when these are broken up. And then finally, you have a more favorable resonance products. So after this phosphate group, gamma phosphate group is removed, it can have more resonance structures in there and be stabilized that way, right? So you have charge repulsion that it's relieving, better resonance, and better solvation. Okay, so those are the main factors in this. And the delta G naught prime for this reaction, just ATP and water going to ADP and phosphate, it is very highly negative. Okay, so that means it's very spontaneous. Okay, and you can use this reaction, the fact that this is has a very negative delta G, very spontaneous, you can use this and couple it to a reaction that is not spontaneous to drive that non-spontaneous reaction forward. Okay. Okay, the actual delta G of ATP hydrolysis differs from the standard state delta G. The actual free energy change is going to depend on the standard free energy, but then the actual concentrations of the reactants in the product. So it does vary a bit. The free energy change is more favorable if the reactant concentration exceeds the equilibrium concentration. All right, so you have higher concentration of ATP than the equilibrium concentration. That's going to add to drive it forward even more. Another thing I should mention that in real life, your ATPs and your magnesium, your ADP aren't really just floating around by themselves. They usually have things like magnesium, calcium, a metal group, a positively charged cation that's associated with them as well to shield those negative charges. Okay, so that those negative charges aren't just out there unshielded by themselves. You do get some shielding with metal cations. Okay. Table 3.5 in the book shows some typical concentrations of these adenine nucleotides in different types of cells. So if you're curious, you don't need to know these values. There probably is a homework question or two that you might have to look up the concentration of these in this table to answer, but you don't need to memorize these. Okay, what you will notice though, the ATP concentrations are generally pretty high. The ADP and the AMP, especially AMP concentrations are rather low, relatively low compared to ATP. And what that does, if you look at that equation again for our delta G at cellular conditions, you're going to have a very high number in the bottom of this fraction for ATP and some small numbers in the top for ADP and inorganic phosphate, which or at least ADP. So that again just helps it drive this forward even more. Okay, making ATP a very potent source of chemical energy. It already is based on its structure, but then the cellular concentrations just really accentuate that as well. Okay. Several phosphorylated compounds have very large delta G naught prime for hydrolysis. Okay, and again, the electrostatic repulsion within this phosphorylated molecule is very strong, and when that phosphate group is hydrolyzed, that repulsion is relieved. Okay. Products are also stabilized via resonance or by more favorable solvation, just as with ATP. All right. Product can undergo further what's known as tautomerization, which then also drives that reaction forward even more, because it it it, it kind of acts as, I guess you could call this kind of the resonance thing. It will after it loses the phosphate group, it will change back into a different molecule and be further stabilized. And that example of that is shown here. There's a molecule called PEP, and PEP is very high energy molecule, extremely high energy. When it loses that phosphate group, right, it that leaves pyruvate, and pyruvate in the enol form. Okay, this is the ene, and that's the ol, enol. Okay, you have a double bond, carbon-carbon double bond, and an OH group. The enol form can tautomerize into the keto form. Okay, in the keto form, this is in equilibrium, but the equilibrium is heavily shifted to the keto form. So if PEP loses that phosphate group, it's the enol form. Okay, the enol form could theoretically, get the phosphate group back there, but because it tautomerizes, it drives it into the keto form, and you don't get that, backwards reaction here. And because of all of these factors, this reaction of PEP, phosphoenol pyruvate, being hydrolyzed, it has the highest delta G naught prime known, at least in biochemistry, negative 61.9 kilojoules per mole. Okay. So if we look at the relative energy of some of these molecules, right? PEP, the phosphoenol pyruvate, is the highest energy biomolecule. Okay. 1,3-bisphosphoglycerate is kind of a close second. Phosphocreatine's up there. Those are all more higher in energy than ATP. Okay? And that's important because these molecules, PEP, 1,3-bisphosphoglycerate, and phosphocreatine, those molecules can be used to make ATP. Right? And then ATP then can be used to phosphorylate other molecules like glucose. Okay. And this is how metabolism works. You have pathways that create very high energy molecules that can be used to create more ATP, and then that ATP then can be used to phosphorylate other molecules or drive other reactions forward. Some other high energy molecules include molecules that have sulfur groups. Okay, and those are also known as thioesters. They have a sulfur group and connected to a carbonyl, which would be a thioester. Okay, and one of those is acetyl-CoA. All right, CoA stands for coenzyme A, a very long molecule. That's its exact structure is a little bit not important to us, so we abbreviate that as CoA, coenzyme A here. But the important part of it is this acetyl group. So you have two carbons with a carbonyl, so a methyl and a carbonyl here, and attached to the sulfur that's on coenzyme A, the end of coenzyme A. And acetyl-CoA is very important in donating acyl groups, so the two carbon groups. Right? So this is how you feed two carbon groups into metabolic pathways. And one of those important things that you use these two carbon groups is the synthesis of fatty acids. Okay. And acyl transfers molecules other than water except the acyl group. Okay. So the last reaction type we'll talk about are oxidation reduction reactions. Okay. Just a kind of a general chemistry level review of oxidation reduction, right? Oxidation reduction reactions are also known as redox, right? The red part stands for reduction, the ox stands for oxidation. These reactions involve either the gain or the loss of electrons. Okay? Oxidation is the loss of electrons. Reduction is the gaining of electrons. Okay? And so that's kind of how I normally remember it is, reduction is losing. Reduction is gaining. So it's kind of a counterintuitive thing there. So reduction is gaining electrons. Oxidation is just the opposite, then it's the losing of electrons. Right? The other way you can think of it is oxidation is something combining with oxygen, and reduction is something that's either losing oxygen or gaining hydrogen. But those definitions are a little bit, I think, harder to remember. Remembering the loss of electrons and gaining of electrons is a bit easier. Okay, why it's called reduction is that when you're gaining electrons, your oxidation state is going down. Okay, so that's the oxidation number is going down. So that's where the term reduction comes from, even though you're gaining electrons. Okay. Organic compounds serve as fuels in biochemistry, and so the fuel source, what's actually being taken out of these fuel molecules are electrons. Okay, and ultimately those electrons are going to be transferred to the main electron transport chain, and that's how a proton gradient is generated, and that proton gradient is then used to produce ATP. Okay, so that's kind of like the last thing we'll learn about in metabolism in this course. But these electrons have to come from somewhere, and they come from basic organic compounds. Okay, and this list of compounds just kind of shows you, at the top are the more reduced compounds, and at the bottom is carbon dioxide, which is the most oxidized. Okay, so things like alkanes would be the most reduced, okay, have the most potential energy in them. Okay, and then in subsequent oxidation states, we see these steps, right? And then ultimately leading to carbon dioxide, which would be the most oxidized form of carbon. Okay. Reversible oxidation of a secondary alcohol to a ketone. So we'll talk about this reaction. We have lactate going to pyruvate. And what happens is, well, you can see, right, we have highlighted in pink here, we have lactate, which has an alcohol group there, changing into pyruvate, which has a carbonyl. Okay, so this is, the alcohol is more reduced, and the carbonyl is more oxidized. Okay, what's happening when we look at this in specifics, we're getting 2 electrons. If you count up the oxidation states in both of these, what you'll notice is there's the oxidation state has 2 more electrons, 2 more negative charges in lactate. And when it changes to pyruvate, those electrons, although they're not necessarily easy to see and follow, they have to go somewhere. Okay, so you're losing 2 of those electrons. And probably more obvious are the H pluses that you're losing, because those aren't in the structure anymore. So those are also being lost. Okay. And those protons are being lost to keep the charge balance. Okay. Enzymes that carry out oxidation reduction reactions are referred to as dehydrogenases. And usually they proceed by a stepwise transfer of a proton, so an H plus, and then a hydride, which is a hydrogen that has 2 electrons. Okay, so then the sum of that would be 2 H pluses and 2 electrons being transferred. Okay. This is a bit of an FYI, but there you can talk about the reduction potentials of certain reactions and rank them in terms of they're in this chart, they're in volts, so the volts of each of these reactions. And so the higher the value for that reduction potential means that has a higher affinity for electrons. Okay. So electrons then are transferred from things lower on this list to things that are higher on this list. And if you look, again, this is a little bit before we talk about a lot of these, but we're going to see these molecules again, things like cytochrome C, the cytochrome C1 and cytochrome B factors, ubiquinone, right? These are all in the electron transport chain, and the electrons are being transferred from things that are lower on the list to things that are higher on the list. And then the ultimate electron acceptor is going to be oxygen, O2, in this chain. And that's what's shown here. It's the highest thing on this list. Okay. You can write a delta E in terms of delta G and the number of electrons transferred and a constant. And this is what we refer to as the Nernst equation. Okay. The way to find this delta E value is you take the reduction potential of the electron acceptor minus the reduction potential of the electron donor. That's how you get the delta E. And this we can kind of rearrange into an equation that we can use in terms of free energy. The free energy equals minus N, which is the number of electrons being transferred, times F, the constant, times the change in reduction potential. Okay. So for a negative delta G, something that's spontaneous, you have to have a positive delta E. So that means the reduction potential of the acceptor has to be bigger than the reduction potential of the donor. Therefore, that's why electrons are transferred from things lower on this list to things that are higher. Some common cofactors that are used for electron transfer in proteins and in enzymes. Okay, NAD and NADP are probably the most common, at least that we'll see. These are kind of unique in that they can dissociate from the enzyme after the reaction. So the reaction will, take place. They'll get these NAD and NADP will get reduced, and then they will leave the enzyme and go transfer their electrons to another molecule. Okay, and so this is the structure of NAD. You have an adenine here. What NAD stands for, I should probably say first, is nicotinamide adenine dinucleotide. So the nicotinamide is up here in pink, And that's really the business end of the molecule. Um, Adenine, we have an adenine base here, although that doesn't really do much in this molecule. And then you have two riboses connected by a diphosphate. So that's where this is where the dinucleotide, adenine dinucleotide is this part, and then nicotinamide is up here. So this nicotinamide, this is in the oxidized form. It'll have a positive charge on this nitrogen. It can accept 2 electrons and 2 protons, and then it turns into this uncharged form. Okay. Which is the reduced form. Okay. Right. And it turns out that the formation of, I should also mention, this is NAD plus, that's the oxidized form. This is what we refer to as NADH, which is reduced form. So NAD plus is oxidized, NADH is reduced, right? That's a lot easier writing, those abbreviations than drawing this whole molecule out. Okay, so we tend to use these abbreviations. Okay, NADP and NADPH are the same exact structure as this, except there's a phosphate group down here. Okay, so those that the addition of the phosphate group just changes what enzymes use that as a cofactor, but they're really their job is the same. Their NAD and NADP are used for carrying electrons. Right. In a laboratory setting, when these are oxidized, reduced to NADH, a change in the UV spectrum occurs, and so you can use that to kind of track how, if you have a reaction that uses NAD as a cofactor, you can use that. In this case, there's a peak at 340 nanometers that grows in as it's reduced. You can use that to track the progress of your reaction. Okay. Another cofactor that's used to transfer electrons is our flavin cofactors. So there's kind of multiple different flavins. Okay, and their structures are shown down here, and so let me go through these. The part that's getting the electrons is up here. It's this three membered ring. We have 3 6 carbon. I guess they're not carbon, so they're just 6 membered rings, but there's 3 of them together. It's what's known as an isoalloxazine ring or head group. Okay, so that's the active part of the molecule. You have that, then it's attached to a ribose sugar and a phosphate group. That's what we refer to as FMN, or flavin mononucleotide. Okay, when that is also attached to another phosphate and an adenine, right? So this is an ADP attached to that ribose. That's what we refer to as FAD, or flavin adenine dinucleotide. Right. Okay, what's going on here? Again, just as before in the NAD and NADH cofactor, this part isn't really that important. Okay, what's important is up here, what's going on, and you're getting electron transfer to that. Okay, what you'll notice is the flavin cofactors can have 1 electron transferred at a time. Okay. So they can accept 1 electron first, and that becomes a dot here. It's a free radical. And what we refer to that as is, in the case of, if this is FAD, this would be FADH dot, or in flavin mononucleotide, we'd call it FMNH dot. So that's one step of the reduction, okay, and we also refer to that as the semiquinone form. Okay, and then you can have another electron transferred, and that becomes FADH2 or FMNH2. That's fully reduced. Okay, why is that important? That it transfers a single electron? Well, that permits the use of molecular oxygen as an ultimate electron acceptor, okay, because you can transfer one electron at a time. Unlike NADH that can be dissociated from a protein, flavin cofactors are tightly bound to their proteins. Okay? And this is just an example of a flavin protein, ETF, which stands for electron transferring flavoprotein. And you can see that the FAD group is right here, and it is very strongly attached to that protein. Okay. All right, so quantum chemistry, this is really just for your information. You're not going to be tested on this. Most organic molecules, including reduced fuels, are in what's known as the singlet spin state. All electrons are paired. They're all in electron pairs. Okay. Molecular oxygen is actually in the triplet spin state, where two electrons are unpaired. Okay. Direct electron transfer from a singlet species to a triplet species is what's known as forbidden, or quantum mechanically forbidden. Okay, that doesn't necessarily mean it doesn't ever happen. It just means it's incredibly slow. Right? And so that's why direct oxidation, the spontaneous combustion of these fuels, biomolecules, doesn't occur readily. Okay? So few cofactors, such as transition metal ions and flavin cofactors, are able to catalyze a consecutive single electron transfers needed for O2 to be able to be used as the ultimate electron acceptor. Okay, flavins are actually kind of cool. So, the reason I kind of show a little bit more of this is because I have some experience researching flavoproteins, but some other things besides metabolism and electron transferring that flavoproteins do, right? Dehydrogenases, that's kind of electron transfer, fatty acid metabolism. But some other things that they do, photolyases, they can repair DNA damage, apoptosis inducing factors, so they can help facilitate apoptosis. Luciferase, which is a enzyme that causes chemoluminescence and illumination, creates light. And then also they're involved in what are known as cryptochromes, which are a family of proteins that are really just kind of a new area of study. They're proteins that are involved in regulating the circadian rhythm, and also in some, like migratory birds, for instance, they're involved in magnetoreception. So the ability to sense the magnetic fields of the Earth. And so these migratory birds have the ability to kind of feel the magnetic field of the Earth and know which way they need to be traveling, depending on what time of year it is, right? And so that's really kind of crazy that this cofactor can do so many varied tasks. Right. So in summary, this is the summary of what we've kind of covered in chapter 13. It's really kind of a intro to metabolism, kind of get you thinking a little bit more about thermodynamics and about reaction types, and also about certain cofactors. that are going to be used. We're going to see them pop up more and more in some of these reactions. Okay, so next lecture, we'll be covering chapter 14, and we're going to be really going into detail in the first metabolic pathway that we'll learn, and that's glycolysis.